Welcome again. Now we are going to discuss the second important control of microbes through physical method, which is radiation. Radiation are basically two types. One is ionizing and second one is non-ionizing. The example of ionizing radiation are X-rays, gamma rays, cathode rays. So these are the example of ionizing radiation. The second one is non-ionizing radiation. The example of the non-ionizing is a single, which is ultraviolet or UV rays. So what is the principle behind these two radiation? That firstly we need to discuss the UV rays. UV rays is a method or the process by which can disinfect the surface of the object. We commonly refer this when we to disinfect the surface of our biosafety cabinets and some areas or some kinds. This UV also use in a water filter or UV water filter we readily use, and biosafety cabinet. So UV rays, we know that UV rays 260 nanometer length is quite lethal to DNA. DNA, we know that the DNA is made up of nucleotides bases, and there is a T nucleotides. So here the T nucleotides, this hydrogen bonds is been split and make a dimerization over here. So this disrupt the DNA structure at the molecular levels, and ultimately suppose that this portion of the DNA has been trapped in a gene. So this gene ultimately devoted to or form the proteins. So if this happen or this thymine dimers has been formed, so this gene inactive and does not make, or the protein which is a functional protein does not make, and ultimately the function of this DNA has been stopped. So this is a mechanism of ultraviolet rays or non-ionizing radiations. The second examples is ionizing radiation. In ionizing radiations, as the names tell, that in this methods proper sterilization has been taken place, and the form of ions, one types of ions which can penetrate the depth of the object. certain objects like antibiotics, cloths, fibers and some plastic materials. So this ionizing radiation can easily penetrate into the depth of these objects and completely sterilization has been taken place. So till here we already discussed about control of microbes by physical methods, and in physical methods we discuss about the first one is heat and second one is radiation. So heat has been divided, we divided into two types, one is dry heat and second one is moist heat. So dry heat we achieve sterilization, proper sterilization, and moist heat again categorized into two: steam under pressure so this steam under pressure achieved sterilization, and second heat in this moist heat are boiling water, boiling waters and pasteurization. So these two, boiling water and pasteurizations, achieve disinfections. In the starting of this lecture we already discussed about the difference between the sterilization and disinfection. Sterilization is a completely removal of all life form of the microbes including the spores, whereas the disinfections, the term where we applied when we remove the pathogenic microbes. So some types of microbes and even the spores has been remains in this disinfection process. And the second radiation is a second physical methods or physical agents by which can we achieve the disinfection as well as the sterilizations. Sterilization has been done by the ionizing and non-ionizing. Ionizing radiation has been used or done by the proper sterilization, whereas the non-ionizing radiations is being only used for disinfection. An example we have already discussed, the ionizing is gamma rays and this one is UV rays. And another method we already discussed, which is mechanical barrier. And in these methods we discuss about filtration, Filtration has been achieved by air filtration or by the liquid filtration, because these two are the heat labile. So, we can sterilize or disinfect by the heats. So, air and liquid, both of them has been achieved by the sterilization. So, in this section or in this lecture first, we discuss about the various physical as well as the mechanical methods by which can sterilize or disinfects the agents or the any areas or the surface. So, this is now the over of lecture 1. Now, in lecture 2 we will discuss about the different chemical agents by which can control the microbes. Thank you.